morning. This video is going to go over the basics of a chromosome. We hear this word all the time when we talk about cells and we talk about genetics, but what really is a chromosome? We're kind of going to look at it from a larger vantage point, and then next week we're going to really dig down deep into the specifics of the DNA and what DNA looks like that makes up the chromosomes. Now, we reviewed the different parts of the cell, so the mitochondria, the Golgi, the ribosomes, and what all those parts do, and we talked about the nucleus, and that's where we're going to focus kind of our attention right now, particularly in human cells, eukaryotic cells. The nucleus is what's going to hold the genetic information. Now, we know that our genetic information is DNA, right? But what is DNA? Well, DNA is this humongously long molecule that has all the information on it that's needed to make proteins, which everything in our body is basically directed or controlled or related to a protein in some way, shape, or form, right? Think of this situation. When you have a set of encyclopedias, right, you have like 26 different books in the set, letter A, letter B, letter C, letter D, okay? If we did not divide up the information in a set of encyclopedias, then the book, the one book, if we made it into a single book or a single unit, it would be so large that, it would be unusable to us, right? Like we would never be able to carry that book of information, or it would take us so long to look up what we needed to in that humongous book of information. So in order to make all that information more usable and accessible, we divide it into sections, the letter A, letter B, letter C. Well, the cell does the same thing with DNA. It's this huge long piece of information. So in order for the cell to be able to use it in the most efficient manner, it's going to divide it into units that we call chromosomes, right? So here's a chromosome. We often think of it as an X-shaped structure, and we'll talk about that in a second. But basically what a chromosome is that it is just a huge wound up structure or sections of DNA. And the DNA is going to be wrapped around structures called histones. So the DNA wraps itself around these structures and it squeezes together it coils and squeezes in order to fit all of this material into the cell. And so each chromosome then is going to be basically a section of DNA. Now, during interphase of the cell cycle, the DNA is going to be arranged pretty loosely, even within the chromosomes, right? It's so loose that when we look at it under a microscope and high power, we can't really see these individual structures. But what happens during mitosis, remember that first phase of prophase, is the DNA is going to just squeeze itself together, and you start to get this visual image of a chromosome structure. So we kind of visualize the chromosome to looking something like this kind of X-shaped structure. Now, the chromosomes has parts. So these long parts here, these linear long structures are called chromatids. Now, depending on whether or not the chromosome has gone through the S phase, a chromosome can have one chromatid, but after it's gone through DNA replication in the S phase of the cell cycle, remember it makes a copy of all the DNA. If it's gone through that, then it would have two chromatids, or what we would call sister chromatids, and these would be completely identical to each other. We also are going to have top and a bottom part. Typically, we look at the length of the chromatids. So the chromatid or the section of the chromatid that's the longest is called the Q arm, and the chromatid section that is the smallest is called the P arm. So these could be reversed depending on the length. At the ends, we have structures called telomeres, which are going to basically help protect the DNA as it goes through some of the various functions that it's going to go through. And then lastly, we have the centromere, which is the center part of the chromosome here. The centromere plays a really important role because not only does it connect two sister chromatids together, but it's going to be the point of attachment during the stages of mitosis and when we talk later this week about meiosis. So remember during mitosis when the metaphase and the spindle fibers formed and it connected to the chromosomes and it lined them all up along the middle of the cell? those spindle fibers are connecting to those centromeres. And centromeres can be located in different places along a chromosome. So there's actually 4 different positions or categories of organization of the centromere within a chromosome. It can be telocentric, acrocentric, submetacentric, or metacentric. So not all chromosomes are going to have the centromeres exactly in the middle, or do they have them exactly like? Different chromosomes will have different placements of centromeres. Now I want to go back to this for just a second. So again, you can see the centromere. We can count how many chromosomes we have in a cell by counting the centromeres. That's kind of the structure that we want to look at. But I want to revisit this idea of chromatids and sister chromatids. So let's say that this is chromosome number 1. We got this one from mom and we got this one from dad, okay? This is what the chromosome would look like before the S phase of the cell cycle. So this is what it would look like during the G1 phase, right? We have chromatid, centromere. We got one of these from dad, we got one of these from mom. After the S phase, when the DNA copies itself, now our chromosomes are going to look like this. They're going to be made up of 2 chromatids, sister chromatids. Now remember, count the centromeres to see how many chromosomes we have. We still have 2 in this one. We still have 2 in this one. But the difference is that we know that this has gone through the S phase or the DNA replication phase of the cell cycle because we have these 2 sister chromatids. So this structure right here is a chromosome, so is this structure. The only difference between the two is whether or not it has gone through that S phase. So this can be confusing to students when they just see this one. They're like, oh, this is a chromosome, and then this is 2, but that's not the case. This is 1 chromosome, everything in green. This is 1 chromosome, everything in green. And so if you get confused, again, count the centromeres. So that's the basic structure of a chromosome, okay? The telomeres, the arms, P-arm, Q-arm, and the centromeres, and then chromatid, either 1 if it hasn't gone through DNA replication, or 2 sister chromatids if it's gone through 2. So the next question then is, what is a gene? Like, we talk about genes, we assume that genes are DNA, but I mean, on a chromosome, what is a gene? A gene refers to a very specific section of DNA on a chromosome. So for instance, the gene for making, let's say, our eye color is found on this location on, let's say, chromosome number 3, okay? On every chromosome number 3 in this section is the gene for eye color. It's not always going to be the same. There's going to be different versions of this gene, but the protein that makes eye color will be found on a specific chromosome at a specific location. And that's what a gene is. It's the specific location on the specific chromosome that codes for the protein that's going to give us a trait, whatever that trait could be. So like here it says eye color, here it says hair color, here it says skin color. It's a little more complex than that, but you get the general idea. So a gene is just a specific section of DNA that's found on a chromosome. So last, we have 23 pairs as humans of chromosomes. We get 1 pair, 1 chromosome from mom, and we get 1 chromosome from dad, 1 from an egg and 1 from a sperm, okay? So here is a picture of a human karyotype, which is just a picture of a chromosome, and we're going to talk a lot more about these during lab. But here we go, we got 2 chromosome A, or 2 chromosome number 1s, okay? So let's just say the gene for hair color was on chromosome 1. It would be like right here, and it would be right here on this one as well. Like it's in the same spot. Different versions, but on the same location, okay? We know that these chromosomes have gone through the S phase because it each has 2 chromatids. Here's chromosome number 2. We got 1 from mom, and we got 1 from dad. Think of these as the individual books in the encyclopedia set. Okay, here's chromosome 3. Maybe the gene for skin color is on chromosome 3. Okay, it will always be on chromosome 3. It will never be on chromosome 10. Okay, chromosome 4. So when we count a human karyotype, one of the first things that we can do to see if there's any kind of problem, developmental problem, genetic problem, is just to count chromosomes. Take a picture and count them, right? We need to have 46, 23 pairs. 23 chromosomes, but we get one from mom and we get one from dad, equaling 46. Chromosomes number 1 through 22 are called autosomal chromosomes because they determine everything about our traits except for sex. Chromosome pair 23 is considered a sex chromosome because it is going to determine all of the traits that are associated with being a male or a female. You can have two possibilities for chromosome 23. You can have an X and a Y. They will be different looking chromosomes. That would give you a male. You can have an X or an X, and that would give you a female. We arrange the chromosomes by size. So the largest chromosome will always be chromosome 1, and the smallest will be chromosome 22. Like I said, 23 is kind of a special situation. So hopefully that gives you a better understanding of what a chromosome is. And we're going to be talking a lot about how we get what happens to these different chromosomes in order to, during fertilization, right? You're having a baby, so we need to figure out what's going to happen. And I want you to think about, before we get to the next parts of these videos or these lessons, is if we have 46 chromosomes in all of our cells, then when a sperm and an egg combine, how many chromosomes would that unborn baby have, or that new cell that's formed, that fertilized egg? And if you have to have 46 chromosomes, 2 pairs of 23 chromosomes, in order to be a fully functional, let's say normal, in quotation mark, human, you can see that we have a problem, right? Because if we have sperm and egg cells with 46 chromosomes and they join together, now we have a cell that's got 92. That's not a human. That's not a functional human cell. So what we're going to be talking a lot about is what has to happen specifically in our sperm and egg cells to cut this number in half so that there's only 23 in each of those cells. So when a sperm with 23 chromosomes joins with an egg with 23 chromosomes, we now all of a sudden have a new human that can develop out of a cell that has a full 46.